Welcome back, everyone. You're tuned into another eye-opening conversation where we explore the transformative world of artificial intelligence, drawing insights from Dr. Alok Aggarwal's groundbreaking book, The Fourth Industrial Revolution and 100 Years of AI.
Glad to be back.
We've covered some really fascinating ground so far, haven't we? From self-driving cars and all the potential and problems they bring, to the incredible world of 3D printing. And then we even talked about 3D printing robots and how AI and big data are changing that world. But today, we are going to shift gears completely. We're diving into something truly revolutionary.
Absolutely. Gene editing and specifically CRISPR.
We're going to uncover how this technology allows us to actually rewrite the very blueprints of life.
Get ready to explore the captivating intersection of CRISPR and artificial intelligence. We'll discuss how this gene editing technology works, where it's being applied, and how AI is revolutionizing both CRISPR techniques and protein structure prediction.
Absolutely. So let's start with the basics. We hear these terms DNA and RNA all the time. But what are they really, and why should we care about them?
So imagine DNA, deoxyribonucleic acid, as a massive library filled with instruction manuals for building and running a living organism. Each book in this library is a gene, and each gene contains the instructions for building a specific protein.
Okay, so genes are like the blueprints for proteins.
Yes.
And proteins are like the workers of our cells.
Well, workhorses, carrying out all sorts of essential functions.
Okay, got it. So where does RNA fit into all of this?
So RNA, or ribonucleic acid, is like a messenger. It carries copies of those instructions from the DNA library to the ribosomes, which are the protein-making factories within our cells.
So kind of like taking a specific blueprint from the library to the construction site.
That's a great analogy. Yeah.
I like that. So DNA is the master plan. Genes are the individual blueprints within that plan.
Yes.
RNA is the messenger.
Exactly. And ribosomes are the construction workers building the proteins. And what's amazing is that all these instructions are encoded in the sequence of just 4 building blocks, or nucleotides: adenine, thymine, cytosine, and guanine.
And those 4 nucleotides, often represented by the letters A, T, C, and G, they are the language of life. And the specific order of these letters determines the instructions encoded in each gene. This is where things get really interesting, because with gene editing, we can potentially change those instructions.
Oh, wow.
Which means we can change the proteins that are made and ultimately alter the traits of an organism.
Now that's where things get really mind-blowing. So gene editing is essentially about going into that DNA library and making changes to the blueprints.
Exactly.
But I understand that gene editing existed before CRISPR.
That's right, it did.
So let's see what made CRISPR such a game changer. Before CRISPR, gene editing was like trying to edit a book using scissors and glue. It was clunky, time-consuming, and often imprecise.
Right?
CRISPR, on the other hand, is like having a precise word processor for DNA. It allows us to target specific genes with incredible accuracy and efficiency. And what's even more remarkable is that this revolutionary tool was inspired by a natural defense mechanism found in bacteria.
Okay, now I have to hear this story. Bacteria with a high-tech defense system. Tell me more.
It all started with a scientist named Francisco Mojica, who was studying these strange repeating DNA sequences in bacteria. He called them CRISPR, clustered regularly interspaced short palindromic repeats.
Wow, that's a mouthful.
It is. It turned out that these sequences weren't just random quirks of bacterial DNA. They were part of an incredibly sophisticated immune system.
So bacteria have their own immune system?
They do.
How does it work?
Imagine a bacterium being attacked by a virus. This bacterium can actually capture a snippet of the virus's DNA and integrate it into its own CRISPR array, like taking a mugshot of the invader and adding it to a criminal database.
Oh, wow.
Then, if that same virus tries to attack again, the bacterium can use the stored DNA sequence to recognize and target the invader.
It's like having a most wanted poster for viruses.
Exactly.
That's brilliant. But how does this bacterial defense system translate into a gene editing tool for humans?
That's where 2 brilliant scientists, Emmanuel Charpentier and Jennifer Doudna, come into play. They figured out how to harness this bacterial system to create a programmable gene editing tool. Their breakthrough involved using a specific Cas enzyme called Cas9 and designing a guide RNA molecule that could be programmed to target any desired DNA sequence.
So Cas9 is like a pair of molecular scissors, guided by this custom-designed RNA GPS. And once this complex finds its target in the DNA, what happens next?
It finds its target and then cuts it.
Okay, so the DNA is cut. Now what? What happens to those loose ends?
So the cell has these remarkable repair mechanisms. They're constantly repairing damage to their DNA. And there are a couple of ways to do this. One way is called non-homologous end joining, where the cell essentially tries to glue the broken ends back together as quickly as possible.
So, that sounds kind of like a quick patch job. Does it always work perfectly?
It doesn't always. Sometimes small pieces of DNA might be lost or added during this process, which can disrupt the gene's function. And scientists can actually use this to their advantage if they want to knock out a gene entirely.
Interesting. So by cutting the DNA and letting the cell do a quick fix, scientists can essentially turn off a gene.
That's right. But there's another repair pathway called homology-directed repair, which is much more precise.
Okay.
In this case, the cell uses a template to guide the repair process. And this is where CRISPR becomes really powerful.
I'm starting to see where this is going.
Yeah.
So if scientists provide the cell with a specific DNA template, they can essentially rewrite the gene.
Yes, that's the beauty of it. By introducing a template with the desired DNA sequence, scientists can trick the cell into using that template during the repair process. It's like providing the cell with a revised blueprint for the gene.
It sounds incredibly powerful.
It is. Let's now explore how AI is revolutionizing the world of gene editing.
Definitely.
So we're dealing with an enormous amount of genetic data. Traditional methods really struggle to analyze it effectively. But AI, particularly machine learning and deep learning, can process and learn from this data in ways that we couldn't even imagine before.
So it's like AI is taking those molecular scissors and making them super precise.
Precisely. Think of it like a super-powered GPS guiding those CRISPR scissors to the exact spot on the DNA, minimizing the risk of errors. And this level of precision is really critical for making gene editing safer and more reliable.
Okay, so that brings us to our first mind-blowing area where AI is changing the game, this hunt for anti-CRISPR proteins. And these are basically like secret weapons that viruses have developed over billions of years to defend themselves against CRISPR.
Yeah, it's like an arms race on a microscopic level.
It really is.
And AI is helping us uncover these hidden defenses.
So how is it uncovering them?
Well, there's this one AI model that was developed, and it predicted over 2,500 potential anti-CRISPR families.
Wow, 2,500.
And some of these have even been confirmed in lab experiments.
Really? So it makes you realize how much we still don't know about the microscopic world.
Absolutely.
But why is finding these anti-CRISPR proteins so important?
Well, if we want to use CRISPR to fight diseases, we need to understand how viruses might fight back. Anti-CRISPR proteins are like
the enemy's playbook. So knowing their strategies can help us design more effective therapies that can outsmart them.
Okay, so we've got AI helping us find these viral defenses. What's another way that it's making CRISPR better?
Imagine this. You train an AI model on thousands of CRISPR edits, both successful and unsuccessful ones. And it learns to distinguish between accurate cuts and unintended ones. It's like having this microscopic quality control expert, making sure that those molecular scissors are always on point.
Okay, so AI is making CRISPR more accurate. But there's another area where it's kind of like changing our understanding of gene editing entirely.
This is a big one.
And this has to do with what happens after those molecular scissors make a cut.
Right? So for a long time, we thought that the way DNA repaired itself after being cut by CRISPR was pretty random.
But thanks to AI, we're starting to see that there are patterns that make some repairs more predictable than we realized.
So AI is uncovering a hidden order in something that we thought was random. That's incredible. What are the implications of this?
Well, the implications are huge. If we can predict how DNA will repair itself, we can potentially influence that process, leading to even more precise gene editing. It's like knowing which way a tree will fall after you cut it, and maybe even being able to guide it to fall in a specific direction.
Okay, My mind is officially blown. We're talking about potentially shaping the very building blocks of life here.
It is powerful technology.
And I mean, from what I've seen in the book, it seems like it can do just about anything. Like we're talking about fighting diseases, changing agriculture, and even bringing back extinct species.
It's really remarkable, isn't it? It's like science fiction becoming reality.
Yeah, no kidding. So where do we even begin with something this big?
I think a good place to start is with disease control.
Okay, yeah, disease control. Dr. Aggarwal mentioned something about CRISPR being used to fight diseases spread by mosquitoes, like malaria.
Right? So you have researchers like those at Imperial College London, for example. They used CRISPR to modify the genes of Anopheles mosquitoes.
Anopheles.
Yeah, they're the ones responsible for spreading malaria. So they targeted the genes that control female fertility.
So the mosquitoes can't lay eggs.
Exactly. They essentially created a generation of mosquitoes incapable of reproducing.
Wow, that's pretty amazing. But wouldn't getting rid of a whole species of mosquito mess things up? You know, like the ecosystem and stuff?
Oh, absolutely. That's a huge concern. Scientists are really carefully looking at that. It's not just about getting rid of a pest, it's about understanding how that species fits into the bigger picture.
Right? Everything's connected.
Exactly. So researchers are looking at ways to use CRISPR for something called a gene drive. It basically ensures that a specific gene modification gets passed down to almost all the offspring.
Ah, I see.
So you could potentially suppress or even eliminate a whole population over time.
Okay, that makes sense. So it's not just targeting a few mosquitoes. It's about making sure that change spreads to the entire population.
Precisely.
Wow, that's powerful. But the book has also mentioned using CRISPR on other animals that spread diseases, right?
Yes, the research goes beyond mosquitoes. They're looking at invasive rodents, for instance.
Oh, right, because they can really cause problems.
Especially on islands where you have a lot of endangered species. Rodents can just wipe them out.
Right, so CRISPR could be a tool to protect those endangered species.
Exactly. It's about giving those native species a fighting chance.
Wow. So CRISPR isn't just a weapon against disease, it's like a conservation tool too.
It is. It's really about using this technology to address some of the biggest challenges facing the planet.
I see a theme emerging here. Okay, so let's shift gears a bit. The book also talks about CRISPR being used in agriculture, which I find fascinating.
Ah, yes, the quest for the perfect cup of coffee.
Now you're talking my language.
Well, scientists are using CRISPR to target the genes that make caffeine in coffee beans.
You're telling me we could have naturally decaf coffee?
That's the goal. Imagine a cup of coffee without the jitters.
Sign me up. But what does that mean for the whole decaf process? And for coffee farmers?
Well, think about it. The traditional decaffeination process uses chemicals, right? And that can affect the flavor and aroma. This new approach, which companies like Tropic Biosciences are working on, could change everything. You could have decaf that tastes just as good, if not better, than regular coffee.
Yeah, it would be a game changer. But it's not just coffee, is it? I remember reading something about algae and biodiesel too.
Right? Algae are amazing for biofuels, but naturally they don't produce a lot of fat, which is what you need for biodiesel.
I didn't know that.
But with CRISPR, companies like Viridos are modifying algae to make them produce twice as much fat.
So they're making them super efficient fat producers.
Exactly. And that makes biodiesel a much more viable and sustainable energy source.
That's incredible. It's amazing how precise these gene modifications can be. But wait, there was something in the book about CRISPR being used to deal with food allergies too. Now that's something I need to know more about.
Well, researchers are looking at how to use CRISPR to change the genes that cause allergies.
Seriously?
Yeah, for example, they've identified the specific proteins in egg whites that trigger reactions, and they're working on ways to edit those genes to create hypoallergenic eggs.
Wow. So in the future, people with egg allergies might be able to eat eggs without any problems.
That's the hope.
That would be life-changing for so many people.
Yeah.
It seems like we've covered disease prevention, conservation, revolutionizing coffee, boosting biofuels, and tackling food allergies. Is there anything CRISPR can't do? This is starting to feel like science fiction.
Well, you might be surprised to learn that scientists are even exploring the possibility of using CRISPR to bring back extinct species.
Wait, are we talking about bringing back the dinosaurs, Jurassic Park style?
Not quite Jurassic Park. Dinosaurs are a bit out of reach for now. However, researchers are looking at bringing back species like the passenger pigeon.
The passenger pigeon.
Yeah, it went extinct in the early 1900s.
Wow. Bringing back a species that's been gone for over a century. That's mind-blowing. How is that even possible?
So they're using preserved genetic material from extinct species and introducing those genes into closely related species that are alive today.
Ah, so they're like using a modern-day relative as a starting point.
Exactly. And then they use CRISPR to fine-tune the DNA, basically trying to reconstruct the genetic blueprint of the extinct species.
So they're editing the genes of the modern relative to make it more like its extinct ancestor. That sounds incredibly complex.
It is. One of the biggest challenges is just the sheer amount of genetic information they have to work with. It's like putting together a giant jigsaw puzzle with some pieces missing.
I can only imagine. And even if they can reconstruct the DNA, how do they actually bring the species back to life?
That's where things get even more complicated. For some species, it might involve cloning techniques. For others, it might be about breeding programs with genetically modified individuals. It really depends on the specific species and what genetic material is available.
Okay, so it's not as easy as just flipping a switch. There's a lot of intricate scientific work involved, but this all raises some pretty big ethical questions, doesn't it? I mean, we're talking about tampering with the very fabric of life.
You hit on a crucial point. The power of CRISPR comes with tremendous responsibility.
It feels like there's a fine line between scientific progress and potentially, playing with fire.
Exactly. And as we explore these applications further, the ethical considerations become even more important.
We've covered so much already. Fighting malaria, engineering algae, and the potential of de-extinction. But we haven't even scratched the surface of the ethical side of all this.
Right? The ethics of CRISPR are a whole other conversation.
Right? Like even with something like disease control, which seems like a good thing on the surface, there are still questions.
Exactly. Like you said, what happens when you eliminate a whole species of mosquito? You could disrupt the food chain. You could impact other species. Even create a space for another disease-carrying organism to take over. Maybe even something worse.
Oh wow, I didn't think about that.
It's a reminder that even with the best intentions, we really need to be careful and think about the bigger ecological impact.
Totally, it's like playing Jenga with the whole planet.
That's a great analogy.
You pull out one piece, and you don't know what's going to happen.
Exactly.
And then there's the whole issue of using CRISPR on humans.
Ah, yes. Now that's where things get really complicated.
I mean, where do you even begin with that?
Well, the book makes a really important distinction between somatic cell editing and germline editing.
Okay, so somatic cell editing versus germline editing.
Right? So somatic cell editing is about modifying genes in non-reproductive cells, like the ones in your skin or muscles.
So those changes only affect the individual. They don't get passed down to their kids.
Right? It's like you're fixing a problem in a specific part of the body without changing the overall blueprint.
Okay, that makes sense. And that seems less controversial than germline editing, right? It's like a more targeted treatment.
Exactly.
But germline editing, that's where things get ethically tricky.
Right? Because germline editing involves modifying genes in reproductive cells like sperm or eggs or even embryos.
So those changes would be passed down to future generations.
Exactly. You're changing the blueprint itself, not just for one person, but for their children and their children's children.
Wow. Yeah, I can see why that raises some red flags.
It's a huge responsibility we're talking about, making changes that could affect people who haven't even been born yet.
And I remember Dr. Aggarwal mentioned a call for a moratorium on inheritable human genome editing.
Yeah, back in 2015, a group of scientists called for a pause on inheritable human genome editing.
So basically saying, hold on, let's think about this carefully before we go any further.
Right. They weren't against basic research or even somatic cell editing.
But they were concerned about germline editing.
Exactly. They urged caution when it came to altering the germline because we still don't fully understand the long-term consequences.
That makes sense. We're still learning so much about genetics and how it all works. And wasn't there some controversy a few years ago about a scientist in China who used CRISPR on human embryos?
You're probably thinking of He Jiankui.
Yeah, that's the one.
He claimed to have used CRISPR to edit the genes of embryos to make them resistant to HIV.
And that caused a huge uproar in the scientific community.
It did.
But why was there such a strong reaction? Was it because the technology wasn't safe enough at that point?
It was a combination of factors. For one thing, there were already effective ways to prevent HIV transmission.
Oh, so it wasn't really necessary.
Many argued that the potential benefits didn't outweigh the unknown risks of using CRISPR on embryos, especially when there were safer alternatives.
Right?
And there were also concerns about the lack of transparency and oversight in He Jiankui's work.
So basically he went rogue.
Essentially, he went ahead with these experiments without the necessary ethical approvals and without fully considering the potential consequences.
It sounds like a classic case of the science moving faster than the ethics.
It was a cautionary tale about the importance of having clear ethical guidelines and regulations, especially when we're dealing with such powerful technology.
For sure. It's like we have this amazing tool, but we need to make sure we're using it responsibly and ethically.
Exactly. Not just for the sake of scientific progress, but for the well-being of humanity.
And future generations, too.
Absolutely.
And Dr. Aggarwal goes on to explain something fascinating, how AI is finally solving the protein folding mystery that scientists have been scratching their heads over for decades.
Yeah, and it's really interesting to see just how much AI is changing things up. I mean, we're talking about unlocking some of life's biggest mysteries here, you know?
Yeah. So let's break this down. First things first. How are proteins even made?
So imagine a bustling factory inside every single one of your cells. And inside that factory, there's a little sac-like structure called the ribosome. And that's where proteins are born. They start as a simple linear chain of molecules like beads on a string. You know, it's a simple chain.
So it starts simple. Then it gets complicated.
It's really complicated.
Yeah, this chain isn't content just staying straight. It starts to twist and turn, driven by forces of attraction and repulsion between the different molecules along the chain.
So like, what's guiding these moves?
It's kind of like magnets. You know how opposite poles attract and similar poles repel? Well, the molecules in a protein chain, they have these positive and negative charges that behave in a similar way. And so those charges are attracting and repelling each other. And that's what drives the chain to fold into that specific, unique 3D shape.
And the book calls this the eventual fold, right?
Exactly.
And this eventual fold is critical because if a protein doesn't fold correctly, it can't do its job.
It's like a key that needs to fit into a lock.
Oh, perfect analogy.
A misfolded protein is like a misshapen key. It won't work.
Exactly. And the consequences can be pretty dire. Misfolded proteins can cause all sorts of problems within the cell, leading to stress and dysfunction. They've even been linked to a whole range of diseases.
Oh, wow.
Everything from diabetes and obesity to even cancer.
So this isn't just some abstract science thing. This is like directly affecting our health.
It's very real.
How have scientists traditionally studied this whole folding process?
Well, for years they relied on a combination of approaches. Computer simulations have helped them model the folding process.
Okay.
But to actually see those protein structures, they've had to use some pretty painstaking lab techniques like X-ray crystallography. But despite over 50 years of research, scientists had only managed to figure out the folds for about 160,000 proteins.
So leaving a whopping 4,800 of them shrouded in mystery.
That's a lot of missing pieces in the puzzle of life.
It is a lot of missing pieces until AI stepped onto the scene.
Okay, so set the stage for us. What happened? What's this AI hero that swoops in and saves the day?
So we've got Google DeepMind's AlphaFold.
Okay.
It's a deep learning network that uses something called transformers.
Transformers.
Yeah, and it's kind of like, think of a detective.
Okay.
Meticulously piecing together clues, finding patterns in this seemingly chaotic case. That's what transformers do. They sift through tons and tons of data, looking for those hidden connections and patterns.
They're like superpowered detectives for the world of protein folding.
Exactly. And the second iteration of AlphaFold, called AlphaFold 2, essentially cracked the code for most of those 4,800 proteins that had scientists stumped for so long.
So they went from like a few pieces of the puzzle to like basically completing the whole picture?
It was huge.
It was monumental, and it earned the creators of AlphaFold 2, Demis Hassabis and John Jumper, a well-deserved $3 million prize back in 2022.
But this is where things get even more interesting, right?
Yeah.
DeepMind kept AlphaFold 2's inner workings a secret. Like, they had this incredible breakthrough, but didn't share all the details right away.
It's like they found the treasure map, but held on to it for a bit before revealing it to everyone.
That's so interesting.
And this secrecy actually spurred another team into action. A group led by Bake at the University of Washington took the bits and pieces of information available about AlphaFold 2, and they created their own AI called RosettaFold.
Okay, so now we have two different AIs.
Yes.
Both tackling this problem, but with their own distinct approaches.
Right. A friendly competition in the world of science, with each team pushing the boundaries of what's possible.
So what are the differences? How do they compare?
Well, you see, AlphaFold 2 seems to be slightly more accurate when it comes to predicting the structures of single proteins. But RosettaFold can tackle more complex structures. It can even predict how multiple proteins interact with each other. And then some scientists even believe that RosettaFold might be better at capturing the essence of protein shapes.
The essence?
Yeah, like those crucial details that ultimately determine how a protein functions.
Oh, that's so fascinating. And here's what I find really exciting. Both AlphaFold 2 and RosettaFold are open source now, meaning any researcher can use them and even improve them.
It's true. It's like sharing the recipe with everyone and inviting them to add their own culinary twist.
I love that analogy.
And that spirit of collaboration is truly at the heart of scientific progress. And so with these powerful AI tools now freely available, the pace of discovery in protein folding and biology in general is going to skyrocket. It's already happening.
That's so cool. Okay, so we've got these incredible tools now that can predict protein structures with, like, amazing accuracy.
Right?
But why should we, the everyday listener, care about this?
Well, think about it this way. Proteins are the workhorses of our cells, involved in virtually every process that keeps us alive and functioning. And when those proteins go rogue, when they misfold, that's when diseases happen. So being able to understand and predict how proteins fold opens up this whole new world of possibilities for treating those diseases.
I see the connection now. It's like we have the repair manual with these tiny molecular machines that are keeping our bodies running.
Precisely. And that's just the tip of the iceberg. This knowledge can also revolutionize drug development. A lot of drugs work by targeting specific proteins in the body, like a guided missile zeroing in on its target.
So the better we understand the 3D structure of a protein, the better we can design drugs that bind to it effectively.
Exactly. The AlphaFold 2 and RosettaFold are essentially giving scientists high-resolution maps of those drug targets, allowing them to design more effective treatments with fewer side effects.
That's huge. And on that note, I think we'll wrap up our deep dive into the world of CRISPR and protein folding. I hope this has given you a glimpse into the incredible possibilities and the challenges that come with such groundbreaking technology.
What a journey we've had today. We've explored the fascinating world of CRISPR gene editing, from its groundbreaking applications to how AI is supercharging this technology. We've seen its impact across different fields and discovered how AI is unraveling the mysteries of 3D protein structures. And here's a thought to leave you with. As this technology evolves, our understanding of it will shape its potential to transform our future.
Exactly. Next up, we're diving into how AI is transforming everything from new material discovery to revolutionizing drug development. We'll explore how it's making breakthroughs in medicine more accessible and affordable, while also taking an honest look at where AI still faces challenges in precision medicine.
And that's a wrap for today. Thank you for being part of this incredible discussion. We can't wait to share more groundbreaking insights with you next time. See you soon.